What is electron transport chain? What protein complexes are used in electron transport chain? How energy currency ATP of the cell is produced during electron transport chain? Electron transport chain is a series of protein complexes and other molecules which transfer electrons from electron donor to the electron acceptor. Before discussing about electron transport chain, first of all, let's discuss about the structure of the mitochondria. Mitochondrial structure consists of two membranes, outer membrane and inner mitochondrial membrane. There is a membrane between these two mitochondrial membrane that is called intermembrane space. If we see the cross section of the mitochondria, we see here that it is the outer mitochondrial membrane and here is the inner mitochondrial membrane and there is a space between these two membranes that is called intermembrane space. Four protein complexes are present on the inner mitochondrial membrane. These are 1, 2, 3 and 4. Now discuss about the one by one complexes of proteins. Complex 1. We discussed in previous lectures of citric acid cycle that during citric acid cycle NADH and FADH are produced that are reducing agents. NADH transfers electron from transfers electron to the complex 1 and FADH2 transfer electron to the complex 2. When NADH transfer electron to the complex 1, it overcharges with the electrons and it pumps hydrogen ions from the matrix. Here is the matrix of the mitochondria. From the matrix to the intermembrane space. Then FADH2 transfers separately electron to the protein complex second protein complex. uh, it does not overcharges with electron as there is less energy uh, in FADH2, so it can't pump hydrogen ion from matrix to the intermembrane space. We see here that uh, NADH as NADH pump hydrogen ion to the intermembrane space. Both complex 1 and complex 2 transfer these electrons to the coenzyme Q. Complex 1, complex 2, complex 3 and complex 4 and other two prosthetic groups which is coenzyme Q and cytochrome C. These are also present in electron transport chain. Then complex 1 and complex 2 transfer to the coenzyme Q. We see here that coenzyme Q transfer these electron to the complex 3. Complex 3 overcharges with electron and it pumps hydrogen ions from the matrix to the intermembrane space. Then complex 3 transfer these electrons to the cytochrome C, which is prosthetic group. Cytochrome C transfer these electrons to the complex 4 of the electron transport chain, which is the last complex of the electron transport chain. It again overcharges with the electrons and it also pumps hydrogen ions from matrix to the intermembrane space. Now, complex 4 of the electron transport chain transfers these electrons to the oxygen molecule. Oxygen molecule splits into two oxygen molecules and hydrogen ions from the matrix combines with oxygen to form two water molecules. Continuous pumping of hydrogen ions from matrix to the intermembrane space and hydrogen ions are collected in the intermembrane space and it creates electrochemical gradient, positive charge in the intermembrane space and negative charge in the matrix of the mitochondria. Then how ATP uh, energy currency of the cell is produced. As we know that the inner membrane of the mitochondria is not permeable to the ions. So to transfer these, to pump these hydrogen ions to the matrix, we need a special transporter which is ATP synthase. It pumps hydrogen ion to the matrix. Pumping of hydrogen ions from intermembrane space to the matrix. Pumping of hydrogen ions from intermembrane space to the matrix of the mitochondria, ATP is produced. ATP is converted into ATP, and this is energy currency of the cell. ATP is produced. One by one, four complexes of the protein. Complex 1. Complex 1, it is also called NADH dehydrogenase complex or NADH oxidoreductase. NADH, it is called NADH dehydrogenase because it removes hydrogen ion from the NADH. Complex means it contains FMN, flavin mononucleotide, and iron sulfur com- compounds. It is also called NADH oxidoreductase. As we have discussed, dehydrogenation is the best example of the oxidoreductase. Complex 1 is L-shaped protein complex. It is present in the intermembrane space and the matrix of the mitochondria. The main function of the complex 1 is to transfer electron from NADH to the electron transport chain. Then complex 2. It is succinate dehydrogenase complex. It also contains iron sulfur clusters. It removes hydrogen from succinate and oxidizes to fumarate. It is part complex 2 is the uh, protein complex which is used in the Krebs cycle. It converts succinate to the fumarate. During this, FADH2 is produced. FADH2 transfer electron to the complex 2 of the electron transport chain. Then complex 3. Complex 3 is called cytochrome reductase. It is also called Q cytochrome C reductase. Cytochromes Cytochrome basically are heme complexes which contain iron core, and it is present in oxidized and reduced form. The main function of the protein complex 3, transfer electron from cytochrome C to the protein complex 4. Then complex 4, it is also called cytochrome C oxidase. It also contains heme and copper units. Complex 4 is called cytochrome C oxidase. Protein complex 4 is used for the reduction of oxygen to the water molecule. We discussed about electron transport chain. First of all, we discussed about the definition of electron transport chain. Four complexes are used: complex 1, complex 2, and complex 3. Complex 1 receives electron, accept electron from NADH, then transfer to the coenzyme Q. Complex 2 accept electron from FADH2, it transferred to the coenzyme Q. Both complexes transfer to the coenzyme Q. Coenzyme Q transfer to the protein complex 3. Then protein complex 3 transfer electron to the cytochrome C. Cytochrome C transfer electron to protein complex 4. Protein complex 4 transfer electron to the oxygen molecule, which is the acceptor of electron. It was all about electron transport chain. If you have any question, you can ask in the comment section. Thank you.